Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm Leah Rosen, Bioprocess International's online editor. Before we get started, just a couple of notes. This webinar is being recorded and will be made available for replay in the multimedia section of our website. We have muted the audio lines for the attendees, but we welcome you to type in your questions for our speaker in the chat window on your screen. Your questions in the chat window will only be visible to myself and our speaker. After the presentation, we will begin the question and answer portion because this is, after all, Ask the Expert, and I will ask our speaker your questions from the chat window. Thank you again for joining us today. It is now my pleasure to introduce Dr. Sebastian Kleebank from Eppendorf.
Hello everybody, my name is Sebastian Kleebank. I am Development and Application Lab Manager at the company DASGIB. Since 2012, DASGIB is part of the Eppendorf Group. Today I would like to introduce our new single-use bioreactor for microbial applications, the BioBLU 1F. On this slide here you see a picture of this bioreactor. The bioreactor has a gas inlet filter for sterile gas supply. It has a fully featured, highly integrated head plate and innovative cooling baffles for high mass transfer and high cooling capacity. Also, this vessel has non-invasive DO sensor port. This means the DO sensor is not in direct contact to the medium because there's a silicon membrane at the bottom. Then we have 3 Russian type impellers in this bioreactor, and there are also versions available with 2 Russian type impellers. There's a magnetic drive assembly for stirring, and for the exhaust condensation we are using a Peltier-based liquid-free exhaust condenser. With this single-use bioreactor we did a 4-fold parallel cultivation using the strain E. coli K12. The pre-culture was incubated using the Innova 42R incubator shaker using LB medium at 37 degrees. Then the pre-culture was transferred to the BioBLU 1F vessels using the DASGIB BioBlock parallel cultivation system. Here we used a minimum mineral medium called pan medium and cultivated at 37 degrees as well. Here on this screen you see the results of the 4-fold parallel cultivation. On the left side on the graph you see the dissolved oxygen concentration, and on the right side you see the feeding pump of our glucose pump. So after inoculation the dissolved oxygen concentration dropped from 100% down to approximately 20, parallel in all 4 bioreactors. This happened due to the growth of the organisms and the increased metabolic activity. Now to prevent that the dissolved oxygen concentration drops below 0, we started our DO cascade. This data is not shown in this graph, but we first increased the stirrer speed and then we increased the oxygen content in the inlet gas stream. We started this run with 40 grams per liter initial glucose concentration. And after this glucose was depleted, you see a sharp increase of dissolved oxygen concentration, a so-called glucose starvation peak. Our software is able to recognize this peak, so we were able to automatically start the feeding in all bioreactors. We started with 14.7 milliliters per hour using a 500 grams per liter glucose solution. And in the course of the experiment, we reduced the feeding rate down to 5 milliliters per hour. On this screen you see the results of the oxygen transfer rates. This graph is an average of 4 bioreactors. In the upper graph you see the BioBLU 1F results. So after the batch we reached oxygen transfer rates of up to 200 millimoles per liter an hour. And after starting the feeding we reached values between 150 and 200. After we decreased the feeding, we also see this in the oxygen transfer rate because the value decreased down to 100. Now for comparison, we applied exactly the same process to our autoclavable glass bioreactors. And on the lower graph you see that the oxygen transfer rate graph is comparable to the graph from the BioBLU 1F reactor. And after we started the feeding here, you see that we reached oxygen transfer rates values between 100 and 150, so a little less than in the BioBLU 1F reactor. On this slide you see the biomass formation during the cultivation. In the BioBLU 1F reactor we reached cell wet weights of 160 grams per liter as an average of 4 bioreactors. The arrow bars on the results indicate that we reached a high reproducibility within the 4 bioreactors, proving that the bioreactor or the single-use bioreactor system can be used for precise process control. And also for comparison, you see the results of the autoclavable glass bioreactor. Here we reached 140 grams per liter cell wet weight, also reproducible within all 4 bioreactors, but on a little lower level than in the single-use bioreactor. Now I would like to give you 2 take-home messages. The first take-home message is that our BioBLU 1F bioreactor is suitable for high cell density cultivations. The stirrer speeds of up to 1500 RPMs combined with the cooling baffles that we have lead to high mass transfer rates. So we could measure KLa values of more than 4000 per hour using the static sulfide depletion method. Also, due to the cooling baffles, we reach heat transfer rates of more than 50 watts per liter. This means we can effectively get rid of the heat, of the biological heat that is formed especially during high cell density cultivations. The second take-home message is that with the BioBLU 1F reactor you can reduce your turnaround time. The bioreactor comes pre-sterilized, so you don't need to clean it. You don't need to, after the process, you don't need to inactivate, disassemble, clean, reassemble and sterilize it. So you save a lot of setup time. When you stop a process, you can start the next one within the same day. So you can increase the throughput of your processes from 1 run per week to 2 runs, and even from 2 runs per week to 3. Another advantage of our bioreactors is that we have a low amount of leachables and extractables. All wetted parts are USP Class 6 certified and made of animal component-free materials. Thank you very much for your attention. I'm now ready to answer your questions, and if you have additional comments or questions, please contact me under this email address. Thank you.
Thank you. So the first question is the oxygen sensor polarographic or fluorescent?
The oxygen sensor is polarographic. So we can insert this sensor into our non-invasive DO port, and it's not in direct contact with the medium because we have a silicon membrane at the bottom.
And what type of sparge is being used, and what options do you expect will be available?
The sparger that we used is simply a dip tube, so we don't use a sinter sparger.
And can you describe the method for PO2 pH measurement? Are there probes included, or do these need to be aseptically installed?
Yes, for DO sensors, we have the polarographic sensor that can be put into the DO port, so you don't need to autoclave the sensor. The sensor can stay connected at our control system, so you don't need to wait for polarization. And when the bioreactor is ready, you can simply put the DO sensor in the non-invasive DO port. Regarding the pH sensor, we are using normal glass sensors, so you can separately autoclave them, and then you can mount them into the bioreactor under the laminar flow using a PG 13.5 port of our head plate.
What is the biggest size of bioreactor?
The biggest size of the microbial version of this single-use bioreactor is the 1 liter that I just showed. The working volume range is from 250 milliliters to 1,250 milliliters. But for cell culture applications, we have also bigger single-use versions available, up to 40 liter working volume.
And do we need the heating block, or is it an internal loop?
For this bioreactor, you need a heating block, so you need to have the DASGIP BioBlock system for cultivation. So you will place the bioreactor in these heating wells, and for cooling you have 4 cooling baffles installed in each bioreactor. And these cooling baffles can be attached to a cooling water distribution unit, so you can effectively cool down the bioreactor.
And are you able to maintain your posted KLA values in your larger vessel sizes?
The KLA values that we determined were in our largest microbial version, the BioBLU 1F. And for the larger vessels that we have, they are cell culture vessels, so you don't need this high KLA values.
And are the units provided gamma sterilized?
Yes, our bioreactors are irradiated. We are using beta irradiation for sterilization. So they are double bagged, so you can unpack the bioreactor under the laminar flow to be sure that it's sterile.
Thank you, Sebastian. Thank you for joining us for this Bioprocess International Ask the Expert webcast.
Thank you.